So we have the great pleasure of inviting or saying hello and invite the first keynote speaker, Professor Dirk Schübeler from Friedrich Miescher Institute of Biomedical Research in Basel in Switzerland to give the first talk. And I'm a great fan of all your work. When I get questions on how is DNA methylation regulated, then we come back to your papers. And I think you have taken on a very, very difficult task, so, but you have done it well. So we look forward to listen to your talk. And you are, I said, you set the stage high, like you have 2 ERC grants over the years. And yes, please, we are looking forward to listen to your talk.
Thank you so much for the kind introduction. Thanks for the organizers to bring us together. I think it's been, for me, also getting back to in-person is really, I think, a big part of what we enjoy in science, actually being around scientists, not only talking about science. So having this again, I think is fantastic. So my lab is interested in the function of chromatin DNA methylation in gene regulation, and as you will see, we've been trying to learn a bit more about the molecular rules, and methylation has been one key aspect. And what I've been trying to do is basically give you an overview, kind of what drives us, and then give you kind of 3 examples of aspects we work on. One is looking at unpublished work. We're looking at how is methylation repressing transcription. Then I'll talk about a new transcription factor, and I'll talk about the dynamics of open chromatin sites when we look at the function of remodeling enzymes. So I think I'm old enough, I was a starting postdoc when the human genome was sequenced, and for those old enough, they know there was a betting going on how many genes our genome would have, and nobody guessed the number being so small, right? It was numbers up to 100,000s out, and it was actually a bit surprised. How could we get away? How can we generate this diversity of cell types with such a limited repertoire on genes if you compare this to the fruit fly, not even twice as many as the fruit fly. And we know now that this diversity relies on one key aspect is that apparently it's been possible to express these genes in all combinations. And this is driven by, as we know, distal regulatory regions. And the beauty of this system is that, let's say you have an enhancer that regulates a gene in tissue A. Because it's distal, this thing can evolve to do other stuff without interfering with this. So it's basically neutral and interferes with the function. And then, we play around, and it's just been and we see that regulatory regions are actually evolving rather rapidly. And we know, of course, at the core of these are transcription factors that recognize DNA motifs. We're still debating how many there are. So it's not as, even though we have many very, very well understood, it's not that this repertoire is fully understood. We also don't have motives for many of them. So, we're still missing even a big part of the sequence-based regulation, something we should keep in mind, particularly those of us that work with chromatin methylation, and we somehow think that genetic part has been somehow solved, and everything we don't understand must be done by this other path. Something we shouldn't ignore is that these components are not all understood. Of course, we know that many disease mutations are within regulatory regions, and we need to understand how they impact that to make any inways into finding disease-causing genes and also understanding or potentially interfering. And of course, we know this regulation happens in chromatin, and this is nucleosomes, this is DNA methylation. I don't need to introduce this to this group. And we know there's many disease-causing mutations in chromatin pathways, so clearly critically to understand. Now, if we look at this interplay between sequence-specific and chromatin, I think one thing we need to understand, I'm going to use this kind of way of illustrating this. One other surprising thing is if we look at prokaryotes tend to have very large DNA sequence motifs that are transcription factors. In bacteria, a transcription factor tends to have a large motif and binds all occurrence of that sequence. If we look at eukaryotes now, particularly in the larger genomes, like ours, the motifs of our transcription factor actually is shorter on average of those as bacteria, even though they have to navigate an order of magnitude bigger space. How is this actually working? If you look at, let's say, this example here, this is looking at C-MYC. This is the site that MYC loves the most, has the highest affinity to. It binds, this is in mouse stem cell, it binds only 2% of these perfect binding sites. And these 2% make perfect sense to us. The guy binds there, there's open chromatin, everything is good, but we really have to explain also these 98%. And of course, we say it's chromatin, but that's just a correlation. What distinguishes these from these? And that's the key in understanding specificity, because also the question is, could this be involved? And we know it's involved in having cell-type specific binding sites. We're going to use this kind of illustration of ability to navigate for different transcription factors throughout the talk. And the idea being, if this number is low, I would just say naively, this guy must be chromatin sensitive. If it wouldn't be chromatin sensitive, it should bind these other, and it doesn't. And keep in mind, it's a bit of a proxy here. We look only at the perfect motif, so we only want to give an idea. It's not really a real kind of mapping of sites. It's a bit more complicated than just looking at the best motif. But it illustrates the point. The other point I want to make, I'm going to give two examples on this, is one thing that, we are getting extremely good at getting these epigenomic maps, right? It's been, methylation, fantastic, single molecule, single base readout. There aren't that many in biology, and that's why it's such a rich data. We know surprisingly little how dynamic these things are. We talk about landscapes. We also use terms that somehow communicate a very strong stability, and of course inspired by body, but we don't know if these landscapes are actually, like the Bernese Alps, or is it like a standing wave that's constantly rebuilt? It's there, but it's key for us to understand. I think it's super important for us to understand how it works. Difficult to study. I think we're missing there a lot, because any concept of stability, of epigenetics, without knowing this, very hard to look into that. I'll give you two examples how this can be addressed, and I think hopefully we can do more as a field. So I'm going to discuss, as I said, unpublished work on how CpG-rich promoters repressed by DNA methylation. I'll discuss a novel activating TF that's published. So I'm going to go a bit farther through this. In the end, I'm going to talk a bit more kind of how do we get to chromatin opening and how it's regulated and how dynamic it is. So this is, again, going back to genome size, right? I mentioned this in the introduction about transcription factors. This is, again, just, if you look at this, if we think about this, it's just amazing, the variation in genome size, right? If we look at, so this in kilobases, so 3 times 10 to the 9, we're somewhere here. Mammals, this is kind of what we're used to. But if you look at plants, if you look in amphibians, right, enormous variations in genome size. So there's plants out there that have, 100 times larger genome than ours. How is this working? Because they have the same problem. In this genome now, there's for the 6 base pair MYC binder, there is now 100 times more sites, right? How is this even possible? Because we know that increase in genome size is largely driven by expansion of repeats. And this is a very nice example. It's been first really very beautiful and inspirational, written by Tim Bester. And here is the largest genome has been sequenced so far, Australian lungfish, published last year. This guy is a living fossil. There's a fossil record of this species 100 million years. During that time, it must have constantly accumulated repeats, because we look at this genome now, and it's 14 times as larger than ours. This accumulation must be happening after this diversion. It's now 90% repeats, but the thing looks exactly the same. So it means whatever this happened here, it must have been able to mask the effects. And we know, of course, it's driven by the expansion of repeats, and it's possible because these repeats are efficiently silenced, so they don't interfere all the time with normal regulation. And DNA methylation is the other thing that I forgot to say. It's also been first noted by Tim Bester and also Lisa shown. From a certain genome size on, every organism has DNA methylation, even in clades where methylation is not mandatory. Of course, we know invertebrates all have methylation, but even if you look at this, and we think it's simply because in order to have a large genome size that's driven by repeats, and the only way to do this efficiently, to do this so many, is having DNA methylation. It's kind of what we think that this correlation holds. So vertebrates are unique in that they methylate all their genome, and this is just a cartoon of these genome maps. I'm sure you're aware, of course, that what is unmethylated normally is CpG islands and active promoters and active enhancers. And this was an observation made 10 years ago by a number of groups, including ours, that these enhancers, there's not many CGs in there, although it required genome-wide bisulfite maps to see this, that these enhancers are actually really dynamic between tissues, and the majority of changes actually happening in this regulatory region. And you can look at them in these regions, and you can see enrichment for certain motifs of TFs that are active in the tissue, and that's cool, has a huge diagnostic potential. Doesn't tell us if it's regulatory or not, and it's something we're keenly interested in the lab. Now, how would this be regulatory? How could the methylation and a few cytosines in enhancer be regulatory? And there's basically the most simple concept would be to say, this is inhibiting transcription factors to bind, or the other extreme, this is driven by transcription factors that are binding. So these are the two extreme examples you can imagine. This is our transcription factor. Here's the site that's unmethylated, and you can imagine this factor demethylates, and this is why we see this. Or you could say, this transcription factor is sensitive to methylation, only binds if it's unmethylated. And I'll give you examples for both, because there are examples for both. There's a huge variety where that's going on. We're still lacking a full understanding. In vitro, there's been a lot of evidence for these affinity differences. Very hard to do this inclusively in vivo, and so we'll be trying to get into that space. So let's look at the first example, transcription factor that doesn't care. REST, or NSRF, this is a transcription factor. It's a silencer. It has a very complex motif, unusual for a mammalian transcription factor, a very large motif. And indeed, if you look where it binds in the genome, it binds 84% of all its perfect binding sites. This guy, according to this criteria, doesn't care about chromatin, finds its motif, binds to it. Very large, so that should work. The same is true for something like CTCF, for example. And if you look where REST binds, again, this is done in mouse stem cells. We see a peak here for REST binding. Here, this is single cytosine methylation. You see this drop around the binding site. Here, look, it's as a cartoon. If we remove REST from these cells, methylation comes back, so REST is required for demethylation. Does REST care about methylation? No, it doesn't. If we put REST back in, it binds again, demethylates again. So clearly, this guy drives a dynamic methylation state and doesn't care. Here's the other extreme, NRF1. We identified this behavior, which has been suggested before in vitro, but we could show this in the cells by using a trick in these mouse stem cells. By removing DNA methylation, we thought, a factor that's sensitive should now explore new sites. And maybe we can grab this by looking at changes in accessibility. And NRF1 was one of the top hits there. NRF1 has this motif, also a pretty large motif for a mammalian factor. And it binds in normal stem cells with methylated genome a quarter of its perfect sites. If we remove methylation in mouse stem cells, it's the only cell system where we can do this, for a reason we're going to explain in a minute, now NF1 binds almost all of the perfect binding sites. So it's clearly indicating this guy doesn't care about nucleosomes, I would say, but it cares about methylation because it's inhibited by methylation of these two CGs in its motif. It's interesting because, according to all criteria, right, this behavior would make him a textbook pioneer factor. This guy doesn't care about nucleosomes. It cares about these tiny methyl groups. And this is important for our view. A binary model of pioneer or not, I don't think does justice to this molecular space, because I'm sure we'll counter all variations of sensitivities that are used in order to direct transcription factors. Now, one can use this now and show that indeed this dependency can be utilized to guide binding sites, right? So REST we know can demethylate. So if there's an NF1 site next to it, we would think REST is required for NF1 to bind here. And indeed, we could show this in this paper that basically, if we remove REST, this site gets remethylated and NF1 doesn't bind anymore. Now that's interesting because it's a molecular principle that basically NF1 now, which is actually a housekeeping factor, binds to present in all cells, can be guided to different sites by TF that demethylates a larger region. Right? And we think that's going to be key to understanding where these things can be regulatory or not. And the beauty of this setup is that this dependency does not require any physical interaction between these factors. It's chromatin, in this case methylation, that acts as a switchboard of this activity that enables differential binding of a factor that is now has now cell-type specific sites mediated by differential methylation. So I'm going to touch on this. We've been asking now a big question, of course, then how dynamic is methylation? And I don't go into this, just more a teaser. If you're interested, look at that paper. So Angelika Feldman in the lab, and they later joined by Paul and Demos. But we basically thought it should be easy to measure turnover of methylation. We just take stem cells, we acutely delete de novo methylation, DMT-A and B, and we look at decay over time. Whoever requires de novo methylation for keeping up, there must be turnover. And then we repeat this experiment in cells that don't have TETs, which is active demethylation, and in theory, that give you turnover rates for each cytosine that you're looking at. In reality, this is a bit harder because you have to really measure it carefully and model this, and you have to have a differentiation, and we never managed to do this in post-mitotic cells. But nevertheless, you get from these turnover maps. Basically, there's places where methylation turns over fast and places where it turns over slow. And the key message is here, high turnover of DNA methylation around transcription factor binding sites. So basically, at these enhancers, this kind of, the dynamics we see between cell types, this is going on constantly. It's getting rebuilt because these machines are working, basically, remethylating, demethylating. It's very stable heterochromatin, right? So these sites here, these TF sites, and we can link it to nucleosome position, but again, if this is interest, and I think this might be ways how to get an insight about how stable these marks are. And in our experience in this cell type, it varies enormously between different cytosines. Not surprisingly, but there's ways how to link this. Also different between different TFs, probably some that recruit TET enzymes or not, but something at least we can explore now. So repression of promoters by DNA methylation, you would think that's solved, right? And we have good models how this works. We know it's as much as it's difficult to know when a methylation is repressive at the enhancers. We know it's a very efficient repression of transcription if it's a CpG-rich regulatory element, right? There's no example of a methylated CpG island being active, right? So it's context matters here again. And I'm going to give you one example of an experiment that I did myself actually many years ago, just to illustrate this can even be done de novo. We've been inserting reporter constructs into this mouse erythroid leukemia cells using a system with Cree recombination. And we did in parallel having the plasmid in vitro methylated all CpGs. So this is genetic. So this gets inserted. Cree doesn't care. Genetically, these two cells are identical. But if you look at expression, so each dot here is a cell and green GFP, this is stably repressed. And this lasts forever. 400 divisions in cell culture. It's not because they lost the ability to transcribe. If you'd force them to demethylate with inhibiting DNMT1, you get reactivation. This is, of course, an artificial system, but it is nice something we've been seeing in the genome a lot. If you are CpG-rich, and this is a CpG-rich construct, maintenance works well, repression works well. We're much more struggling in interpreting methylation effect at CpG-poor sequences. Now, how does it work? And there's basically two models, which are not mutually exclusive, and the idea is actually they work together, is that one is direct repression, a transcription factor like NRF1 is blocked directly because the motif is methylated. The second one, of course, is indirectly by proteins such as MBD domain proteins that recognize methylated CpGs and thereby recruit co-repressors, and of course these things of course could work together. There's alternative modes of repression, which I'm not going into, but we kind of wanted to look into these two aspects. And of course, mammalian MBD proteins have been identified, pioneering work by Adrian Bird. They do have an MBD domain. They bind methylated DNA in vitro, in vivo, no question. And it's these four guys that are established to be the CpG methylated readers in our genomes, MBD1, 2, 4, MECP2, the causal gene for Rett syndrome, highly studied. And as I said, binding in vitro, in vivo, fully established interaction with co-repressors, so it all makes sense. What has been lacking is actually doing a genetic test of this model, and this is what we've been done. One thing I also should say that in that context is that there's also a counterpart, which I'm sure you're aware of, is so-called CXXC domain proteins that are actually recognizing the unmethylated CpGs, and there's over a dozen of those in our genome. So Sebastian and Sylvia teamed up for this. We're both former grad students in the lab. And our idea was, let's genetically delete all MBD proteins. That should remove this pathway. And let's contrast this to cells that lack all DNA methylation. That should take out both pathways, and then we should contrast this and should learn who does what. So Seba basically, and I can tell you why this is possible in mouse stem cells. They're the only cells where we can remove all methylation and it doesn't interfere with viability. It's because they are at the developmental stage very early where the genome is largely unmethylated and they still have a pathway that's methylation independent to repress repeats, via recognition of repeats via Krebs zinc finger proteins leading to K9 methylation. This is why we think they can cope with not having DNA methylation. So Seba did a consecutive CRISPR, different strategies, different order to make sure that there's no clonal effect. And these cells are viable in culture, they look normal to us. If we look at transcriptome, there's hardly any change. And the genes that do change, we cannot link this to methylation of the regulatory region. This is different if we look in the DNMT triple knockout, no DNMT1, 3A, and 3B, no methylation. There, and this has been described before, several hundred genes go up, and among these are genes that have a methylated CpG island, such as a class of germline-specific genes and others that go up. So this was a surprise. Of course, you might say, well, why would you do this experiment in ES cells? You know already they can cope with not having DNA methylation. Why should MBDs play a role? So, of course, we would have wanted to do that experiment also in differentiated cells. Now, the problem with this is that these DNMT-KO don't differentiate in classical differentiation paradigms, right? They die along the way. Many have tried, including us, and so we tried to come around with a workaround introduced by Christiane in the lab in Sevi. And the idea was, can we make neurons that lack methylation if we do a forced, rapid program? And this is basically adapting approach that's been developed before. It's an inducible expression of a neurotrophic factor, NGN2. Doesn't matter. All what you need to know is that within 48 hours you make neurons. Of course, this is not looking at development. It's a forced system, but it's looking now GFP labeling of NGN2. And you see in this culture, within 48 hours, very synchronously and you know, without much cell death, we make neurons. Of course, we not looked at neurogenesis here, but we can create now neurons. And these are, you know, people who they express neuronal genes. You know, you can patch clamp them. These are neurons, and we can make now DNMT triple knockout neurons and look at post-mitotic cells that don't have DNA methylation. So if we do this now, basically, and we look again at expression, what we see is that this is a gene that has a methylated promoter in neurons, SZ1. And we see it's only active in the DNA triple TKO. These blobs here are the exons. No reactivation in the MBD quadruple knockout. This is true also genome-wide. There's a few more genes going up and down in the MBD, but again, we cannot really link this to methylation of regulatory regions. We see now again massive changes in the DNMT TKOs, and again, we see these genes like this one here, where the promoter is methylated and reactivation is happening if we remove methylation. Now, is this limited to this mouse system and in vitro differentiation? So we wanted at least create the same knockout in human cells, and Seba did this in HEC293 cells. A transformed line that has many methylated CpG island promoters. And again, just looking at absence by Western, and again, we see in transcription there isn't much happening. This is one guy, a germline-specific guy that's driven by a methylated promoter. If we force demethylation by azacitidine, this guy goes up again. We see no reactivation in the MBD quadruple. Are there changes in chromatin accessibility, right? And again, I mentioned this in passing, and when we looked at NRF1, the logic here in this experiment we do in the lab many times, remove a pathway, look at changes in accessibility as a proxy for potential difference in TF binding, look computationally for motif enrichment, and nominate potential factors that respond to presence or absence of certain chromatin pathways. It's a nomination, of course, but then you have to really follow up. So let's look at the accessibility landscape in these neurons that lack MBDs or DNA methylation. And again, almost no change in the MBD, massive changes in the DNMT TKO neurons. Several thousand of regions go up. There's also some that are reduced in accessibility. If we look at motif enrichment using a tool developed at the FMI by Michael Stadler's group, Mona Lisa, basically looks for motif occurrences, we again find NRF1, that's reassuring. But we also find guys that we haven't found before, such as HNF6. There's also motifs here that don't make sense to us because they don't contain a CpG. So this is clearly not just a list we have to follow up, and we followed up at least one guy here, HNF6. HDF6, also called 1-cut1, is, and now we can show, is methylation sensitive, but only at a certain motif variant. Its perfect motif is this guy, ATT-GAT, which it binds to. Not all occurrences, but many. If it also binds ATC-GAT, but only if this site is unmethylated. This is the methylated site, no binding. If we look in the neurons without methylation, now this site gets bound, so clearly this guy is responding, and I think that's why we kind of keep expanding the list of TFs that are responding to differential methylation. Now, I told you that we can make ES cells that lack methylation because they can repress repeats. And in differentiated cells, that's the job of demethylation as far as we know. And also these neurons that lack methylation, they don't last so long in culture, so they clearly have problems. So we expect, of course, that repeats are getting upregulated in these neurons, as has been shown for in vivo deletions of DNMT1 in the brain. And indeed, that's the case. So repeats are massively upregulated in these DNMT TKO neurons. This is looking at which fraction of RNA contains any repeat signal. And you see this goes way up in the DNMT TKO. Sorry, this is the green, this is lacking here. Not much happening in the MBDs. And it's really driven by a certain class of repeats in mouse. The most recent expanded repeats are IAP retroelements, which are very efficiently repressed normally, but then go through the roof in these neurons that really drown in repeat-derived RNA. It's like a couple hundred-fold upregulation. We can see now which repeat families these are, but that's really not the point. But again, how could this be happening? Because it doesn't happen with MBDs. Is it direct repression of transcription factor binding? So we looked into this, and it's difficult because these are, of course, repeats and much harder to align to really know which unique sequence is involved. Just to show you the structure of these 5' 3' LTR GAG-POL-M gene of these retroviruses. When we look bioinformatically, we see that those that do respond to not having methylation tend to have this CRE element, CAMP response element, that is 1 CG. So Seba tried to recapitulate this first at this genomic landing site, inserting an LTR with CRE and without CRE driving luciferase gene, and having a housekeeping promoter driving luciferase as a control. This is a PGK promoter. Put them in these ES cells, make neurons, look for activity plus minus DNA methylation. And what you see is that the viral containing sequences are very efficiently repressed, are recognized as repeats in the ES cell, become methylated, stay repressed in normal neurons. In the DNMT2 neurons now, these LTRs are highly active. They're de-repressed, and they're twice as active if they contain the CRE element, suggesting that indeed the CRE element is contributing to LTR activity. Which guy binds? Now there's a couple of TFs that could bind this element, and so after some failure, we homed in on CREP1 and basically did chip for CREP1 in wild-type and TKO neurons, and basically the overlay of these tracks shows you that there is increased binding of CREP1 in the TKO neurons. If we now delete CREP1, we see reduction activity. It's not going down to zero, but it's clearly a significant contribution. Also, accessibility goes down. So we really think that also this is driven ultimately by transcription factors that respond to methylation. So basically, our model is that, that blocking of methylation sensitives right now, it's the one path where we get signal for. In these cell types that we looked at, in these mutants that we looked at, doesn't mean that MBDs don't have this function in other cellular circumstances or during development. Clearly, right now, this is the pathway for which we get signal. And we're just trying to see, ultimately, at some point, it doesn't make sense to me. I think I really expected this to be functionally contributing. So that's why I think that maybe a big contribution could be also not the blocking of CXXC domain proteins. That's also happening between these two states, and something we've been now starting to look into. Yeah, so basically, because that would explain why, because not all TFs have CPGs in their site, right? So that cannot be as simple as that. But this is right now the path where we clearly get signal for. So let's move to the second part, and this is just, again, this published work, but I think it's It fits nicely here because it shows that indeed our set of TFs, our motif set, is even not complete. And this is work by Ralph and Lucas. Ralph now, meanwhile, started his lab in Heidelberg. Lucas is a bioinformatician in the lab. And it started as a bycatch of a different approach, which I don't have time to explain. We looked at the ability of motifs to recruit factors. And we included also so-called orphan motifs, which there aren't that many in our genome. Orphan motifs, we use this term for motifs where there's some signal that this sequence does something, but we don't know who's binding. Usually it's the other way around. We know what the TF binds, and then we look for that motif. This sequence is called the CG element, has been shown in reporter constructs to drive expression. And nobody knew who was binding. And we basically, Arno Krebs, now group leader at EMBL, when he was in the lab, introduced a footprinting approach that was initially developed by Michael Kladde into the lab, which basically uses a methyltransferase that likes to methylate DNA in vitro, so in nuclei, and it doesn't do it so well if there's a nucleosome, and it doesn't do it so well if there's a transcription factor. And what it does, it gives you a footprint of occupancy. It only methylates GC, not CG, GC, so you can use it and then use bisulfite sequencing. This is how it's done. Again, nuclei incubation, again introduced by Kladde, and then from Peter Jones moved to genomics. And what Ralph did, he inserted several hundred sequences, and we only talk about one here, into our genomic landing site with a motif, and then did footprinting. And this is if you do a scrambled motif, some average accessibility because there's nucleosomes at various positions. If we introduce a rest motif, remember that's the guy that binds 85% motif, it gives a killer footprint because this thing is binding here, and basically almost all molecules are protected by this TF bound. And if you now introduce the CG element, you also see that there's a strong footprint. And that, of course, got us excited because it means there's something binding here. We have a readout of binding. We should be able to try to catch which guy this is because we have a readout of its activity. Now, Ralph did the obvious, did an oligo pull down in, again, all mouse stem cells with this motif, and there was one hit coming out, band P. We looked at this and we thought, no way that this is meaningful. Usually get hundreds of things pulled down, and we looked at this thinking, no way. Particularly if we looked at what was known about band P, it didn't make any sense because there was a few papers on this protein supposed to bind the nuclear matrix, be a silencer, was officially annotated to not be a transcription factor. This bend domain, of course, there have been links to DNA binding, but everything being repressive. So you would have never touched this guy out of 100, but since it was one, Ralph went ahead, and it turned out to be the right one. So BAN-P indeed binds the CG motif, and this is just a chipsec for this binding very precisely. It binds mostly in CPGI promoters, has 2 CG, so this is where the motifs are. If the motif occurs in islands, it binds all the time. If the motif occurred distally in CG poor sequence, it binds about half of them, which already got us in the suspicion that maybe also this guy is responding to DNA methylation. Here's a motif that's not bound in wild-type ES cells and methylated, and of course we did the usual trick. We did the same experiment now in TKOs, and then you see that this number goes up. So indeed it goes to more sites. The unmethylated genome, this is true in ES cells. This is also true if you look in human cancer cells. If you look at variations in methylation at BAN-P sites, it nicely follows binding. In this case, we can also show this in vitro by purifying the protein together with Nico Thome and Alicia. We can show that the affinity to the unmethylated motif is higher than to the methylated one, actually 12-fold or something, 7-fold, sorry, difference. Less than clearly higher affinity to methylated than to scrambled. What's even more interesting, this guy, at least if you just look at the motif output, is a super strong activator and had been totally missed. I think that's some lessons here and surprises, right? How can we miss a housekeeping, a transcription factor that drives housekeeping genes? How did we miss this guy? I'm going to get to this in a second. But if you just take 3 motifs of BAN-P and you drive luciferase, the same approach we did with the LTR sequences, put this in the CS cells and measure activity. And we do the same for NF1, GAPA, SP1, all 3 strong established CpG island TF driving TFs. This guy is doing much, much better. These 3 motifs do give you a third of the activity of the entire PGK promoter. So we think it's a really strong ITF, and this is something Ralph is working on now to figure out how does this guy is such a strong activator. So of course we would have loved to do loss of function analysis with these things, and BAN-P turns out to be essential for cell survival. We never could recover knockout clones. You know, it's also listed as pan-lethal in these genome-wide knockout screens in cancer. And maybe that explains why it was missed, because when you touch it, a cell dies, so it's hard to really pick this up into any genetic thing. So to work around this, we applied then the DTAC system, which is basically adding a peptide to your protein of interest, similar to the Oxen system, but this in our hands is much stronger. In this case, using the system introduced by Jay Bradner's lab that then use this compound to drag it to ligase and then to degradation. And Ralph tagged the allele, and then in about 30 minutes of DTAC, we don't see any protein. The cells die within a day, so it all makes sense. But of course, we can study now what's happening in absence of the protein. And then basically here, this is a BAN-P side. This is the track of transcription, the exons of this gene. This is a tubulin cofactor. And basically, if you particularly look at intronic signal, because that's, of course, the fast responding signal, right? This is the one that has high turnover. In our hands, looking at intron signal is as good as doing SLAM-seq. And then you can look at the degradation at the absence of transcription, the absence of BAN-P. And if you do this exon intron analysis using this approach, again developed by Michael Stadler, you see very fast loss of intronic signal at bound genes, unbound no effect, and exon, of course, with the usual delay. Really, most genes that are bound by this guy also respond to his absence. Is really a strong activator. It also, and this is important for our thinking in the lab about CpG islands, I always thought CpG islands are open chromatin, right? They're K4-methylated, even when not active. They're the kind of sunny part of the genome. Now, in the past, we thought there are not much nucleosomes. Now, with better sequencing technology, actually, we see them very clearly. They are as nucleosomal as other sequences. And we can even see, like for BAN-P, right? Here's a chip for BAN-P. Here's the nucleosomes, or ATAC-seq, peak around BAN-P. And if you delete BAN-P within an hour, this thing breaks down. It becomes inaccessible. And we see also that nucleosome position now becomes, again, changed. Again, the guy opens chromatin, phases nucleosomes. This is part how it activates. We can, again, now use the system for differentiation into neurons. I'm just going to go through this quickly because it's published. Basically, we can make neurons, again, deplete BAN-P, same effect, strong downregulation of activating genes. And there's also neuron-specific targets, which are also depending on this guy that is inactive in neurons, but driven by BAN-P in ES cells. And again, it's linked to methylation of regulatory regions. Again, a factor that is sensitive to methylation, and there are a few incidences where differences between cell types can be explained by differential DNA methylation of its binding site. Just to summarize this part, so I think the key thing is here that there are still TFs to be identified, and that's something for me that was surprising. It's also surprising to me that this is so exquisitely sensitive to methylation, because when we published NRF1, the way we've been looking for NRF1, we looked for an extreme behavior of methylation sensitivity. So we thought, oh, this is a nice molecular example, but it's a huge exception. And now we find the same behavior in the TF where we was not even part of our question initially. So we wonder if there's basically more out there that are highly sensitive. And our working model right now is that indeed island activity rise on methylation-sensitive TFs. With these activators that are sensitive, also with the lack of effect of MBDs, it's one path, of course, I would like to explore more experimentally to really say, is this how it works? And at the last part, I want to quickly only touch upon another aspect the lab works on, which is getting to the space of cofactors, right? If a TF binds to open chromatin, right, then we see this open chromatin, right? Nucleosomes have been moved compared to this state, so we know ultimately this requires remodeling enzymes. And that's a different world of molecules. There's different families of remodeling proteins. They then have different subcomplexes, so very complex biology. We know they're super relevant. So SNF, of course, we probably know, most mutated family in cancer. And but of course, at some point, to make hypersensitive sites, you would need to move nucleosomes. And so we basically got into this by comparing SNF family with the SY family by making knockouts of the individual catalytic subunits, BRG1 and BRM for this, SNF2H for this family. And the result, just in a nutshell, is that to our surprise, and what we saw is that there are transcription factors that are dependent on SNF2H4 binding and creating open chromatin. CTCF, for example, being sensitive to SNF2H. So in the knockout, reduced binding, reduced accessibility, no response in the BRG1 knockout. And TFs that are highly sensitive on BRG1, such as REST. Why was this surprising? Because it was specific for these TFs, and these are TFs that have very large motifs, so we would have thought that maybe they don't need any helpers. Even they rely on remodeling enzymes. Now, a big question, of course, was, how does this work, right? Is this one thing that in the nucleosome gets moved, this piece of concrete that gets moved, the TF binds, and then forever is open, or is there some dynamic turnover? And there's, of course, different scenarios. You could do, maybe it's continuously required, maybe it's only required once for establishment, maybe once for every cell cycle. And again, it gets to this point of turnover rates, right? And we don't know this, but I think it's important for any concepts of these accessibility maps, how they're generated. Now, we had one important tool we could use. Our institute is core-funded by Novartis, so we have close ties to Novartis, and they have basically developed an inhibitor, small molecule inhibitor of BRG1, BRM, that's the catalytic subunit of SNF. And we basically use this inhibitor, and first could show that the inhibition recapitulates the genetic deletion. So basically, this is mimicking the knockout. So now we can use this, throw it in, and ask over time what happens. And the result basically is that upon inhibition of BRG1, we see very rapid reduction of accessibility. This is looking, for example, here at a certain track of a sensitive TF, and you see within 10 minutes the signal drops, right? And we can look this over time. We can do washouts and ask the system revert back. And this is really where compounds have a huge possibility to look at dynamics and establishment of patterns. Here we just look, for example, this is OCT-SOX, of course, famous pluripotency factor in ES cells. Within 10 minutes, we get a huge drop in accessibility that doesn't go down further. These guys don't care about BRG1. They also don't respond. CTCF also doesn't respond to longer treatment. If you now wash this out, again, you get rapid reintroduction. So clearly, the TF can again go in and open the site. And we can see this for REST, for OCT4, for ESRB. Also has been shown for the OCT4 requirement for this by Freeman et al. And it shows that basically these systems are, again, forming very dynamically, right? Basically, this chromatin accessibility, similar to methylation, even much faster than methylation in my view, is established over and over again. It's not moving a piece of concrete out of the way, or if it is, it's something you have to do over and over again to maintain this. So rather than having kind of the idea of chromatin opening as a door, we use this analogy of suggesting it's a revolving door. You have to do this over and over again to keep a site open and to ensure accessibility. Same results using inhibitor as well as degradation have been also shown by Schick et al. in back-to-back with us. And I think it's important also, in the concept of this conference here to really see, as useful as these epigenomic maps are, diagnostically really understand, we still are only at the beginning to understand, are they reflecting ongoing activity or are they indeed instructive? And there's not a simple rule here. It's different. In my view, it's different for different factors. It's different for different sites. We just need to have to work our way into this and look, in a certain context with this TF in that sequence. It might be instructive or reflective or a combination of things. And it's really something where there's a lot to be learned. So let me summarize. What I showed you is this surprising result to me that we don't see evidence for, in the system we tested, of deleting these 4-MBD proteins in the repression of CpG-rich promoters. We still think that CXCC proteins that bind, again, also the CG, could play a role as being blocked from binding. So we don't think it's just complex motifs or facts that are sensitive. This is right now with the signal we're getting. Again, I showed the example of this TF we missed, and the big question here is, to which I don't have an answer. Are there more out there? I wouldn't be surprised if there are more out there that work on CpG islands, simply because even though CpG islands are, as you probably know, 2/3 of our promoters are CpG islands, because they tend to be the more active genes, 80%, 4 out of 5 times RNA pol2 initiates in our body, it's at a CpG island. We know surprisingly little how they're regulated. And the reason is because they don't work well in vitro. All these great systems that have been developed in yeast, in the fly system, in vitro, to look at these events of initiation of transcription, these don't work well with islands. Plus, people have looked mostly at developmental specific promoters that are mostly CpG poor. So we really lack a deep understanding how CpG islands are regulated. And I think that's something we'll be exploring more into that, because I think it's been simply be a bit missed, and it might be a bit harder. For example, all these islands have no Tata box. There's no TBP involved. We don't even know where the start sites are. And if you look at the start sites, can actually wobble around. So they're quite messy. And given how relevant they are, maybe there are more out there. I don't know. We'll try and look, see. And I gave you an example of this high turn of accessible genomic sites. Again, limited to a certain remodeler, limited to those TF that respond. But I think it shows that maybe also here, these systems are cranking faster than we think, and maybe a lot of these differences we see are actually indeed generated de novo all the time, which of course is important to distinguish and ask how much of this is actually information that can be integrated over time versus just a reflection of ongoing activity, is something we really need to work out. And again, there will be, in my view, there will be all sorts of possible combinations. We just have to really try to find ways how to measure this. So with that, let me thank the people in the lab. I mentioned them along the way in these projects. Great group of people. We're always looking for new people in the lab. Closely collaborating with Michael Stadler at FMI and our functional genomics proteomics imaging core, and with Nico's lab at the FMI. Michael van Baron's lab has been working with us on the BAN-P projects. I think these guys for funding, Novartis for core funding, and these grant agencies. Thank you for your attention. I hope there's time for questions. Thank you very much again for the invitation.
So thank you very much. I think you answered questions, but you also gave us new questions. We will come home and think much more. So this great talk is now open for discussions and questions, please.
Fantastic talk. What I was just wondering is what proportion of human CpG islands have this BAMF-P motif?
It's actually only a couple hundred. It's also not much there because it has 2 CGs adjacent. With this CG decay over evolutionary time, there isn't that much of BAMF-P sites outside of islands anymore. Of course, this also must have worked early when there were more CGs. So it's not thousands of genes. It's a couple hundred. It's essential genes, so basically that's sufficient as a phenotype. Yeah.
Thank you.
I see one question there and one up the back. You are not allowed to say the question before you get the microphone, because it's also all the people on the...
So, Hi. So first of all, great conference. Thank you very much.
Thank you.
And well, I would like to ask about the idea of the transcription factors being cause for the changes on DNA methylation.
Yeah.
So in our group, we haven't seen that in the regulatory regions there are high rate of SNPs that actually can impact, change the binding of the transcription factor, and that in turn, I think that it's associated with that idea. So I would like to ask, what do you think it's causing, or a pathway, or generating these changes from the transcription factor to the modulation of nearby CPG sites? Thank you.
Yeah, so I mean, so we've been studying this a bit for REST, and it's been actually complicated. So in REST, we can take out, remember REST is the guy that demethylates, we can take the guy out, and now we thought it's easy, now we just bring back REST in different pieces, and we figure out how it does that. And we can now separate this, we can separate binding from chromatin opening, from demethylation, but to really work our way up molecularly to the binding partners, which is really what you want, the function we can separate. Now, what we do right now is, we tend to find in these assays TFs that are very good in finding their motif, right? And we don't find 6 base pair binders here because they can go to somewhere in the genome, bind, make a hypersensitive site. At regulatory regions, we get another complexity. There's a lot of small guy binders that work with each other, right? Regardless of methylation, even in any promoter. So now the cause-consequence question, the dynamics, the requirements to deal with nucleosomes, all this stuff, become, of course, much, much more. You can think about all scenarios. Reducing the affinity a bit for one TF, might block the other guy from moving that nucleosome, which might lead to methylation. So the real game is, I agree with you, is at regulatory regions, at promoters. But there you have multiple guys binding, and how we separate this is super, super difficult. That's why we tend to, by definition, find those guys where we can interpret the signal, and it tends to be the guy who can, on his own, bind somewhere and get an anti-CP. The real action is actually these 6 base pair guys, much, much harder. Even without methylation, just in the context of any kind of nucleosome.
Thank you. And then another question at the back.
Hi, super interesting talk, thanks. I'm still getting over your MBD quad knockout results back here. You mentioned when you knock them out, CXXC proteins might come in, but
No.
or CXXC domain proteins might come in?
No, they don't come in. Sorry, I meant, because based on the MBD result, I would not throw out that having methylated CG alone has an impact. And of course, the reader of that could be blocking a CXXC to bind. Because we and others also have shown adding unmethylated CGs to promoter always helps. Right? There is a signal there. Our data would say it's not the repressive activity of MBDs, but it could be keeping CXXC away, right? And again, it would be methylation sensitivity, and it doesn't what I wanted to say, and I wasn't probably clear enough, it doesn't have to go, just because we don't see MBD doesn't mean it has to be all complex transcription factor motifs, right? It could be then the CG alone still, but then you would postulate it being via CXXC.
And
Okay, we take one question here, and then we go to the virtual.
We can talk later.
Yes.
I was wondering if you had a model. Do transcription factors work with specific chromatin remodelers, or are they free agents?
Yeah, so in terms genetically, we see dependence, right? Very clear dependence. It's been very difficult, and that's true actually for most cofactor spaces. We know these TFs converge onto a very limited set of cofactors, activators, repressors, right? We know it has to be specific. There's actually, biochemically, to link, to pull down, see the guys you interact with, there's some few examples, has been very, very difficult. It's limiting us. If you just pull at these TFs and ask, is my remodeler in this? You rarely see a signal, but you rarely see a signal anyways. It's true for most cofactors, we lack this. Now, many of these cofactors, this is another thing we work on, is that there's, of course, enormous batteries of chromatin sensing domains, PhD, bromo, what have you. No TF has these, but these cofactors have them enormously. So there could be a combination of chromatin and direct interaction that we cannot recapitulate in a pull down. It's a big, big bottleneck for us to move on towards the business end of transcription factors.
Okay.
Do I need a mic, or is that?
Probably.
It's on already. Okay, so we've got one online question. I've got a question, so I'll do the online one first, because I can ask you over coffee if there's not time. So Ken Murakami has a complex question. I'm trying to understand this. So he has a question about the first part of your talk. He says, if DNA methylation can block some transcription factor binding, these transcription factors cannot cause the demethylation of their binding sites. In this scenario, what factors determine demethylation of these DNA regions?
Yeah, we would postulate it in the cases we studied by a TF that can demethylate, and these footprints are wider. Now you can, of course, lose methylation passively, so we don't know. And we know a CpG island is recognized in being shouldn't be methylated, right? So basically, if you throw a CpG-rich sequence into an ES cell or other cells, even if it's from E. coli, the cell says you should be methylated, you're CG-rich. There's some sensing system that sees that.
And I'll quickly ask
you have, there's 2 minutes and 43 seconds.
2 minutes, 43 seconds.
For one question,
it's
so
about the
let's go.
timelines, really, because, I mean, it's great work, and you're showing these rapid effects. And you sort of show that at an hour, for instance. And how far can you push that? Can you experimentally look before an hour? Do you have any sense for how rapid is?
For the reduced accessibility, that can be in 10 minutes, within 10 minutes. But it doesn't go, I should also say, simplified it, it doesn't go down to 0, right? You would say, so it's clearly more complicated than that. I think it only says, and this is important, that we just don't look at these things as being static, right? And you might say that's not even surprising. Why would you think that in this molecular dynamic space? But in terms of our thinking about this and the way kind of some things are phrased in the literature, is like, you move this thing out of the way, off you go, development. And I think we just have to be a bit more, it could still be that the initial event requires more energy than later, all these things, but to think about as some concrete you move out of the way, true for methylation as well. It's very, very, very site-specific. And the good news is, the exciting part is we have no ways to look at that in a way, but I think we have to get to this kind of component to study the temporal axis also in the molecular space.
Thank you.
Can I ask? You will have to take it after. I'm here. I'm just thinking, for all of us doing EWOS, looking at differences between disease. Did you have the same question?
Maybe.
Yeah. That we look at diabetes versus control, for example, and then we have difficulties replicating our findings. Because say we're looking at thousands of people, and we do one cohort and then we do the next, and you have these rapid changes. And basically, do you think these rapid changes make it difficult for us that when we're looking at the people, they are actually different? Because we're just taking this second, we're looking at the sample.
I mean, these results are all compatible with, which you guys already see, DNA sequence influences in the first place, because you might influence the TF binding site or not. That's clear local. Second, of course, timelines as well, right? So, and again, I think that's the big question, right? I mean, there is this enormous diagnostic potential of these things because they respond. But when are they instructive? How much is this canonical? I think that's on one side, we need more molecular understanding, and then I guess from the genomic side, we need larger cohorts to get sense for signal strength and to relate signal strength to more sequence variations and things like that.
Yeah, but the question is if the DNA methylation is more dynamic than we think, based on what you have said.
Yeah, but I mean, I think it's important, right? Actually, it was one of these meetings here where we first learned about this subtle change in methylation that people were seeing with these bead arrays. And for me, it looked at this, it didn't make any sense, but it was real, right? There was a 2% difference, it was predictive. But molecular, this didn't make any sense. This is how we got into enhancer methylation. Because then suddenly it made sense, because now, if you would have a genius population of cells, and a subpopulation goes from 2 to 5% where this thing is methylated, you would see that 2%. If you're good enough in measuring this, and these bead arrays are, you would see it, right? So the signal is real, but at least for me, then it made sense how you could get these variable sites, why there would be, for example, maybe reflecting cell type composition in peripheral blood, or why they would be responding to other things, because the signal was real, but we had no molecular concept. At least with enhancer methylation, then it could, with easy molecular, imagine how this arises. Doesn't mean that it is, how stable it is, how instructive it is, different question.
Yeah. Great. Bas, you have to take it in the 5 minutes, whatever the break was called. And now we are 1 minute over time. So we have a 4-minute break, and then we are back. And thank you very much.
Thank you.
Wonderful talk.